plus in the chat, please, if you can hear me, so I know you're all here. Hi. This cycle will be the death of me. No, we're going to make it really easy. We're going to simplify it right down so you'll be absolutely fine. Don't worry. Can you put a plus in the chat if you can hear me? Hi. Hi, everybody. Hi, thank you. Okay. So I'm hoping you can hear me. Brilliant. Hi everybody. If you're on TikTok, if you're watching over on TikTok, do make sure to come over to LauraDoesBiology on YouTube, because you'll be able to join in with the chat and ask your questions and things like that, which will be much better. Okay, so today's stream, we're going to be focusing on the light independent reaction, which is the second stage in photosynthesis. Obviously, all of these streams have been about important cycles in biology. So this is our fifth stream and our fifth cycle. At the end of today's lesson, I'm going to talk to you about the course that we're starting in January for all of you A-level biologists that want some extra help with your exam preparation and your revision. So I'll talk to you about that at the end of the lesson. For now though, some of you I know won't have been to one of these free lessons before. So it's always nice to just get to know a little bit about you. If you could type into the chat and tell me where you're from. I recognise some of the names already. Celeste, I think you've been here before. Recognise your name. If you're new, let me know where you're from. Let me know what you're studying, or even what topics you find the hardest in A-level biology. Just type that into the chat for me. I'm going to tell you a little bit about myself just to get started. So if you don't know, if you've not been to one of the streams before, my name is Laura. I'm head of biology at my EdSpace. I have been teaching now for 16 years. I'm originally from Manchester, if anyone else here is from Manchester. Now I live on the Wirral, which is just near Liverpool. Amelia's from Liverpool, nice. I went to Manchester University. Loved it, did biology. As well as teaching, I've done a lot of work with the exam boards in terms of examining, marking exam papers. So hopefully that's going to help you with your exam technique. And when we come to focus on past paper questions, I've got a lot of hints and tips that I can share with you to make sure you get all of the marks. Yeah, so we've got people from all over. Few people from London, Oxford, Windsor, lots of people from down south, Sheffield, Liverpool, Bristol. Oh, already got some topics, yeah, so you don't like photosynthesis. Well, hopefully we're going to help you with that today, because we're going to break down the Calvin cycle, make it really easy for you to understand. Respiration, I don't know whether you were here last week, we looked at the Krebs cycle last week. Which exam board? This lesson is going to be applicable to all A-level biology exam boards. AQA, I've also taught OCR in the past, so I know what is required for OCR. And to be honest, photosynthesis is one of those key topics, which is on all of the exam boards. And the light independent reaction that we're doing today, the content is really the same for all of the exam boards at A-level. So it's going to help all of you. Struggling with essays. Yeah, actually the reason I chose to focus on cycles in biology is that's one of the essays that has come up a few times now on AQA biology. The importance of cycles in biology. So obviously the Calvin cycle is something you could talk about in an essay. as well as the Krebs cycle. We've done the cardiac cycle, we've done the breathing cycle. So that was actually why we chose these topics for these free stream lessons. Okay. Thanks for all your comments. Do feel free to keep typing into the chat if you've got any questions. Just type them in there. Basically, that's the main rule for these free streams. The rule is you ask as many questions as you like, type them into the chat, get involved, get engaged, and try to get the most out of the stream as possible. So there's no silly questions, just ask away. Either myself or one of the moderators in the chat will answer your question. So we do have some moderators in the chat room that are going to help me to make sure all of your questions get answered, whether that's about the content today or whether it's about the course that we're launching in January. Just make sure you ask all of your questions. Today we're focusing on an A-level topic, Hussein. It's A-level biology, so we're focusing on the Calvin cycle. So yeah, definitely an A-level topic today. As always with these free lessons, you are able to get your study notes. So if you stay till the end of the lesson, you will be able to access some professionally designed study notes that have been written alongside my presentation today. And as I say, that the aim of this and all of my lessons is to simplify the complex content, make it as easy for you to understand as possible. And the study notes really reflect that, okay? They're really easy to follow. We've got some professionally drawn diagrams that are much better than a lot of the diagrams you see in textbooks or revision guides that can sometimes overcomplicate some of these processes, okay? So make sure you stay on and get those notes on the light independent reaction. Okay, let's get started. We're going to start with this from GCSE biology. As I've just said, the lesson isn't focused on GCSE. It is going to be focusing on A-level, so we're going to go into a lot more depth and detail. However, we're going to start with this because this is what you do at GCSE, and it's an overview of photosynthesis. So it is very simplified. We've simplified the whole of photosynthesis into one chemical reaction, which you'll all recognise from GCSE. But in reality, photosynthesis is much more complex than this. The overview is just telling us what reactants we use and what products we make. So we've got the reactants over on this side of the equation. We've got carbon dioxide and water reacting together, and we've got the products over here, which are glucose and oxygen. We always write, don't we, at GCSE, we write on the arrow light energy and chlorophyll, because we also need energy from light and we need chlorophyll, which is in the plant cells, in order for this reaction to take place. But this is an oversimplification, so we're gonna go into lots more detail. As I've said, it's actually a two-step process, and today we are focusing on stage 2. We've put this image in. This is a cool guy. This is Melvin Calvin. What a name. Melvin Calvin actually won a Nobel Prize for his work on photosynthesis. The Calvin cycle, or the light independent reactions, were named after Mr. Calvin. That's why we call it the Calvin cycle. He basically pieced these reactions together from his research on plants in 1961. So he won a Nobel Prize for that. Let's have a little look then at where photosynthesis takes place. So you will recognize this organelle. This is obviously a chloroplast. Now, you will have studied the structure of a chloroplast in A-level biology when you did cell structure. You will have gone through all of the organelles. and you will have learned about the structure of a chloroplast, but let's just go through it again. If you have a look with me over here, the chloroplast actually has a double membrane because it's got both an inner and an outer membrane. So together, we call that the double membrane. Just like a mitochondrion also has a double membrane around the outside. Inside the chloroplast, we have the stroma, which is kind of the fluid-filled centre of the chloroplast. So we've coloured that in yellow to highlight that. You've then got these discs, which are called the thylakoids. So each one of these discs here is a thylakoid. They actually stack on top of each other. You may know that a stack of thylakoids is called a granum. Okay, so a granum is a stack of thylakoid discs. Now, in terms of locations for these reactions, stage 1 photosynthesis, which is the light dependent stage, happens in the thylakoids, but we are focusing today on stage 2. Now, stage 2 takes place in the stroma, so this fluid that fills the chloroplast. Someone just asked me for the definition of stroma. You wouldn't be asked to give a definition because it's really just like a space. It's a fluid-filled space in the chloroplast. In terms of a definition, it's important that you know it's the site of the light-independent reaction. That's what you would say. It's where the light-independent reaction takes place. Why is it called light independent? I'm sure you all already know it doesn't need light, not directly anyway, okay? It doesn't directly need light. However, later on we are going to talk about what would happen if there was no light, okay? But this stage, which remember is stage 2, does not directly need light, hence the name light independent reaction, or if you've seen it called the Calvin cycle, that's because Mr. Calvin discovered this set of reactions. Okay, let's have a look at the cycle itself. Can you just let me know at this point who has already studied the light independent reaction? That would be really useful. So who is here just to revise it? Who hasn't done it yet? Let me know in the chat, please. Okay, that would be useful. Remember, if you are watching over on TikTok, because we're live on TikTok as well at the moment, if you're watching over there, come over to LauraDoesBiology on YouTube, because it would be great to see you in the chat and have your comments and questions. Okay, Amber, you have someone saying not yet. I have. If you're in year 13, you probably have done this already because it's one of the first topics that you do, isn't it, in year 13? Some of you saying not yet. Okay, photosystems. Someone's talking about photosystems. We're not actually going to learn about photosystems today because photosystems are what we talk about when we do the light-dependent reaction, Angel. Photosystems are on the thylakoid membranes. I mean, they're embedded, they're dotted around photosystem 1, photosystem 2, but we're in the stroma today, so you need to get yourselves inside the chloroplast, into the stroma, because we're doing the second stage, which is the independent reaction today. Okay, so it's a cycle. And we're going to go through the stages of this cycle and break it down and make it as simple as we can, okay? Actually, I'm just going to go back to that previous one. We're not going to talk about carbons yet. So let's start here. So we start this cycle with a molecule called Ruby P. Now, you can call it Ruby P but it does stand, let me see if I can spell it. I'll tell you what it stands for. It stands for ribulose bisphosphate. That's what it stands for, ribulose bisphosphate. But you can shorten that in your answer to RuBP. So that's where we're going to start the Calvin cycle. Nearly said carbon cycle then. That's a topic for another day. So we're going to start with RuBP. And as you can see, that RuBP is going to join with a molecule of CO2. Now remember, this is in a plant, so carbon dioxide diffuses into the leaf through the stoma, the stomata. It will go into the chloroplast, it will go into the stroma, and carbon dioxide is going to join with RuBP. Now that requires an enzyme, and whether you're doing OCR or AQA biology, you do need to know the name of the enzyme. Okay, so the enzyme here is called Rubisco, and it catalyzes the reaction between carbon dioxide and RuBP. Now at this point, I'm just going to put up the next slide because you may have noticed it actually had some little green circles. Does anyone know what those green circles are representing? Can anyone work it out? I'm not going to tell you yet, but see if you can figure out what those green circles are for. Okay, whilst you type that into the chat, I'm going to carry on explaining the cycle. So we've got carbon dioxide joining with RuBP. Now we can call that carbon fixation. We say the carbon is being fixed. We can also call it carboxylation. We are carboxylating the RuBP. Yeah, loads of you getting that answer right. Carbons. I've showed you here with the green circles, I'm showing you the number of carbons in each of these molecules. Well done. Because it is handy to keep track of the carbons. Where does the light independent reaction occur? We're in the stroma of the chloroplast. Okay, so in the chloroplast, in the stroma, that's where this is happening. So carbon dioxide joins with RuBP using the enzyme Rubisco, and that's what we call carboxylation of RuBP, or carbon fixation. That is going to produce two molecules of something called GP. GP actually stands for glycerate 3-phosphate, but you can just call it GP, okay, which makes it a lot easier. Now you can see that each GP has got 3 carbons. So that makes sense, doesn't it? Because we've taken 1 carbon from the carbon dioxide, we've joined it with RuBP, which has 5 carbons. That will make a 6-carbon compound, which will split into 2 3-carbon GPs. Okay. Now the GPs, the 2 GPs, will then get converted into 2 molecules of triose phosphate, or TPs. We got GPs and we got TPs. Okay. So you can say that two GPs are converted into 2 TPs, or triose phosphate. But if you have a look at my arrows here, we do need a couple of really important substances for that to happen. Now we need ATP. As you know, ATP provides energy. So we're providing the energy to convert those 2 GPs into 2 TPs. We also need NADPH, because that's going to provide the hydrogen which is needed to convert GP into TP. Someone tell me in the chat, do we know what ATP stands for? Can we remember what ATP stands for? If you were here last week, I think we actually looked at ATP and the structure of ATP last lesson. So type in the chat if you know what ATP stands for. But ATP provides the energy and NADPH provides the hydrogen, because this reaction is an example of reduction. Remember, when you add hydrogen, that is reduction. Reduction is gain. Gain of hydrogen is an example of reduction. So NADPH provides the hydrogen, and that hydrogen reduces the GPs into the TPs. Yeah, good. Adenosine triphosphate. Well done. Adenosine triphosphate is our ATP. It's got a ribose sugar, an adenine base, and 3 phosphate groups. Well done. Okay, so now we've got 2 triose phosphates. Triose phosphate, the plants can then use to make glucose. But remember, glucose has 6 carbons, which you can see with my green circles. Triose phosphates only have 3 carbons each. So you will need 2 triose phosphates to make 1 glucose. You can't use these 2 immediately because a lot of these triose phosphates are going to be needed to regenerate the RuBP. So you can't use, if you use those 2 and straight away make a glucose, you're going to have no triose phosphates left to regenerate the RuBP. So I'll give you some numbers and we will go over this later, but 5 out of every 6 triose phosphates actually go towards regenerating RuBP. Just so the cycle can continue and not stop, you can only use 1 out of 6 triose phosphates to go towards making a glucose. Okay, you can't just use those 2 straight away to make 1 glucose. Give me the formula for glucose then, please. In the chat, you should know the chemical formula for glucose. You should also be able to draw it at A level. You should be able to draw it. Obviously, I can't ask you to do that in the chat, but you can definitely type in the formula for glucose. Let me just read that question there. How is glucose produced if there are only 6 carbons from GP6? So to make 1 glucose, you would need the cycle to happen 6 times. We're going to go over some of that maths later, but yeah, this would have to happen 6 times to get enough triose phosphates to make 1 glucose. Yeah, all of you getting that right. In terms of your glucose, C6H12O6. Well done. Who is confident they could draw that? You don't have to do it. You can't do it in the chat, but just let me know who is confident they can draw glucose. Alpha and beta, you've got to be able to draw both. Okay, let's go through it again. Now, these are on your study notes. So if you stay till the end, you're going to get this really straightforward, simple diagram, as well as the straightforward notes. Now, these notes are sufficient to get you full marks on an extended response question, okay? Without overcomplicating it. Like, I know a lot of the videos I've watched online, or a lot of tutors that do revision, do complicate it, and you end up panicking that it's so hard, but it doesn't have to be. That's my kind of whole approach to teaching. Keep it simple. You can understand it, okay? I don't want any of you thinking, oh, it's too hard for me, because then I wouldn't be doing my job properly. So let's go through the steps. We've got RuBP combines with CO2. We call that carboxylation. So RuBP is carboxylated. Uses the enzyme Rubisco. You need to name that enzyme, okay? It forms 2 molecules of GP. Those 2 molecules of GP are then reduced into 2 molecules of TP, or triose phosphate. But remember, that step requires energy from ATP, and because it's a reduction, it does require hydrogen. Gain of hydrogen is reduction. So that hydrogen comes from NADPH. I just want to stop for a second and have a look in the chat. Alpha glucose has OH below and beta above. Well done. Celeste, ABBA. Celeste, I love that. I've not heard that before. ABBA, that is spot on. Alpha below, beta above. ABBA, I'm going to use that. I'm going to steal that, Celeste. Yeah, the difference between alpha and beta. Alpha glucose has the OH group below carbon 1 and beta glucose has the OH group above carbon 1. So that I'm going to steal that, Celeste. That is really good. Okay, going back to the maths. Remember, most triphosphate has to be used to regenerate the RuBP. So this is the regeneration of RuBP. So in fact, 5 out of every 6 triphosphate has to be used to regenerate RuBP. Otherwise, you'd run out of RuBP and the cycle would stop and then plants would not be able to make glucose. So only some triphosphate can go towards making a glucose. It's 1 out of every 6 goes towards making a glucose. But remember, we do need 2 triphosphate to make 1 glucose because glucose has 6 carbons. So we need 2 triphosphate, 2 lots of 3 carbons to make 1 glucose. Let's just flash this up for a second. We started off the lesson with this. This was our overview of photosynthesis, but I said at the start, it's oversimplified. Now, we're looking tonight at the light independent reaction, and what we've seen so far, we have seen where we need carbon dioxide, and we've also seen how we make glucose, okay? So if you think back, we need carbon dioxide to carboxylate RuBP. So we saw that right at the start of the Calvin cycle. We said RuBP was carboxylated. That's where plants need carbon dioxide. We've also seen how they make glucose. They need 2 lots of triphosphate to make a glucose. Okay. We've not seen water and we've not seen oxygen. We've also not seen light energy or chlorophyll. That's because everything else shown in this reaction is required for the first stage of photosynthesis. So stage 1, which is light dependent, needs chlorophyll and light energy as well as water, and it produces oxygen in the light dependent stage. Can I go back to the last slide? Let me just put it back up here. So that was the overview. Remember, this will be on your study notes. So thinking about our GCSE equation, we've got carbon dioxide here. That's where it's needed. It joins with RuBP using Rubisco. And we've got glucose down here. We make glucose using 2 lots of triphosphate. Okay. Let's continue. Let's go through. This time we're going to fill it in together. Please feel free to go into the chat and just type in. I'm going to do it in this order. Okay, I'm going to number them. I'll number them here so that if you want to join in on the chat, if you're feeling confident, you know what number, what order I'm going to fill them in, and you can type into the chat. Give me your suggestions before I write it in. What is number 1? What is number 2, etc. Okay, so I'm going to start here with number 1. Do type it into the chats. I know there's a slight delay, but type it in, see if you can remember what is going to go here. It is a cycle, so we could start anywhere, but I always start the same place just because it's easier to remember it if you start at the same each time. Okay, I'm going to write it in now because I can see that lots of you are putting it into the chat. So we've got RuBP, ribulose bisphosphate. Yes, and we've got number 2, carbon dioxide. So RuBP is carboxylated. I'm just going to add, actually, I'm going to add the name of the enzyme again. So it uses Rubisco, which is the enzyme we need. Has anyone got number 3 in there yet? Yeah, perfect. So number 3, we're going to make 2 lots of GP, which is glycerate 3-phosphate, but don't learn the name, just call it GP. Number 4, well done. We've got ATP, which we require for energy. We've also got NADPH, which don't forget we need for the hydrogen, because this reaction is an example of reduction. We are reducing the GP. Number 6, yeah, we've got that as well. Number 6, we're going to make 2 lots of TP or triose phosphate. And number 7, a few of you've got that right as well, we've got glucose, which is the whole aim of photosynthesis. That's the whole point. Remember, year 7 biology, you said plants make their own food. We're not going to say that anymore, but plants can make glucose, which we are now seeing exactly how they do that. It's one of the reasons I love A-level biology. Like, you actually get to learn how these things work. You think back to year 7, you just said, yeah, plants make food, plants make glucose, but now you can actually learn how they do that, which I think is cool. Guys, from that glucose, they can make loads of other stuff, you know. They can make starch from glucose. They can make cellulose from glucose. They can make lipids from glucose. They can even make amino acids from glucose, because glucose is basically carbon, hydrogen, and oxygen, right? All of these other compounds need carbon, hydrogen, and oxygen. So they can build all of these other biological molecules using glucose as the building block. They can break it down and use it to make all of these other things. Okay, so when you get your study notes, that will be blank. So I'm going to suggest that you fill that in on your study notes as revision. I'm going to show you some questions. Have a little read through these questions with me. I'm hoping that you notice something about these questions. Have a little read through, type into the chat, what do you notice about these questions? We've got describe the light independent reaction, describe how plants make glucose, describe how plants use carbon dioxide to make organic substances. Does anyone know what I'm trying to get at with each of these questions? They would all be worth about 4 or 5 marks. Yeah, they all start with the same command word. They all start with describe, which probably the nicest command word, isn't it, on a biology exam? Because if it says describe, you know you've learnt it. You should have learnt it in class, and you should, if you've done enough revision, you should be able to get all the marks for a describe question. What else do we notice though about these questions? Describing process, yeah, exactly, precious. They are all asking you the same thing. All of these questions are taken from past papers, and they all mean the same thing. Describe the Calvin cycle or the light independent reaction. So don't panic and think, what does it mean, make organic substances? They're all just different ways of asking the same thing. Okay, so you would answer them in exactly the same way. You just need to realise what you're being asked there. Okay, they're all AO1 because they're all describe. I hate application questions, yeah. Application questions are going to be harder, but remember, they can't ask you an application question that you can't have a go at. You must have learned something in your lessons that you can apply to that question. So that should give you confidence. You shouldn't have to have like a total stab in the dark, because that would not be fair. You've got to think, what have I learned in lesson that they could be trying to get me to suggest here? Because you will know something. okay? You will know something. So Celeste, Asterix, have confidence in yourself. You know enough, you can tackle those application questions. Okay, let's put our answers into the chat for question 1. That is a super quick 1 mark question, but you do need to be precise. So type your answer to question 1 in the chat. Question 2 is for you to have a go at. Again, if you're feeling confident, type your answers into the chat. You can use bullet points. In your exam, use bullet points. So many students ask me, can I use bullet points? Of course you can use bullet points. It's not an English exam. We don't have to write in fancy paragraphs. Use bullet points. In fact, the examiner marking your paper will love you if you use bullet points, because that's what the mark scheme will look like. The mark scheme will be bullet points. And if you hit those marking points in the same order, bang, 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 the examiner is going to love marking your paper. I would always use bullet points. Okay, we've got the right answer to question 1. So we've got stroma. Be a little bit careful. If it says which organelle does it take place in, obviously we'd say chloroplast. But if it says be precise or be specific, we're going to say stroma of the chloroplast. Okay. Describe the light independent reaction of photosynthesis. So this is, like I said before, it's the same question we had on the screen before. They've asked it in all sorts of different ways, but you're just describing the whole process. Let me go. I think I've got... Here we go. This is a model answer that I have written for you. Have a little read through. Yeah, a couple of questions about bullet points then. Yeah, they can check your quality of written communication, but that does not mean you cannot use bullet points. You can still use quality of written communication. Isn't asking, it doesn't care about the length of your sentences, or it's focusing on the scientific terminology that you use and the order that you write things. So when it says quality of written communication, you can still use bullet points. They're just going to check, have you got the process in the right order, and have you used correct scientific terminology? It's not assessing you in the same way as they would say in English. So it's about using the correct keywords, spelling them correctly, and structuring your answer so it flows. So in fact, bullet points can help you with those quality of written communication marks. Celeste, in the essay, you can still use bullet points. I wouldn't use just bullet points in an essay. So what I'd do with an essay is I'd introduce my paragraph. For example, if I'm talking about cycles in an essay, I'd introduce my paragraph by saying, the Calvin cycle is an important cycle, the light independent stage of photosynthesis. But then I could go on to then use bullet points to describe that cycle. As long as I use the right keywords and my bullet points are in the right order, you can use bullet points in an essay. Elizabeth, Malika, I'm loving your answers. Well done. So we've got Ruby P joins with CO2, uses Rubisco, makes 2GP, GP reduced to TP, uses energy from ATP, don't forget to say energy, using the hydrogen from NADPH. Most trisphosphates used to regenerate Ruby P, but 1 out of every 6 can be used to make glucose, which can then be used to make other organic compounds. I also said at the bottom there, it might be worth saying this happens in the stroma, because that might get you an easy mark if you say it happens in the stroma. Okay, so application questions. I know someone said earlier they hate application questions, so I'm going to throw a couple at you. But I'm going to show you what I mean by, if it's an application question, you do know something. You must know something that you can apply to it. They're not going to ask you just a random question. So this one, for example, if a plant was subject to very high temperatures, what would be the effect on the light-dependent reaction? Sorry, that's meant to say light-independent reaction. Doesn't matter. Light-independent reaction. Now, this is not quite the same as just asking you to describe the cycle. Obviously not. They're making it harder. They're talking about something called heat stress. But you know the cycle, and you also know how enzymes are affected by temperature. So put your suggestions into the chat. If a plant is suffering heat stress, what do we think would be the effect on this cycle, on the light-independent reaction? Now, I've already seen an answer there that is spot on. What if we said this was worth 4 marks? We'd have to extend on that a little bit. We've got Rubisco will, Astrox, Rubisco will denature. Rubisco will denature, but that's only going to get you 1 mark. So Rubisco will denature, you can see I've put an X through it there. It will denature, but let's carry on. Let's imagine this was worth 4 marks. If Rubisco denatures, can't catalyse the reaction between RuBP and CO2, therefore no GP can be made. Brilliant, we're getting there, aren't we? So, if Rubisco denatures, RuBP cannot be carboxylated, which means we cannot make the GP. And if we can't make GP, we can't make the TP. And if we can't make the TP, we can't regenerate the RuBP. Yeah, and you're right, we'll make less glucose. Okay, so do you see what I mean? It's an application question, but you have learned something that they want you to give as an answer. So you're taking your knowledge of enzymes and how they're affected by temperature, you're taking your knowledge of the Calvin cycle, and you're putting it all together. Okay, let me just show you my answer to that question. So I've said the rate of the light-independent reaction would be reduced, Rubisco would be denatured, less RuBP combines with CO2, less GP, less TP, less RuBP. So you're basically ending up describing the cycle again. You're just saying less of it's going to happen because the enzyme is going to start to denature. Okay, which means its active site is going to change shape and it will no longer be complementary to its substrates, which in this case are RuBP and CO2. Let's have a look at one more. Again, not as straightforward because we're linking different things together. So we're being told that carbon dioxide is a limiting factor, or often a limiting factor. In fact, you should know there's only about 0.04% carbon dioxide in the atmosphere, which for plants, it often does become the limiting factor. Now, again, how are we going to answer this question? Celeste, RuBP is carboxylated. What that means is it joins with CO2, but that requires an enzyme. So if I write it like this, this is carboxylation. So we're carboxylating RuBP, joining it with CO2. But for that to happen, we need Rubisco. Rubisco catalyses that reaction. It brings RuBP and CO2 together and reacts them together. So you need Rubisco for that carboxylation. Okay, so this question then, very similar again, taken from a past paper, very, very similar to the previous one. It's almost worth, if I were you, in my exam, I'd be doing a quick little sketch of the Calvin cycle, you know, just just next to the question, I'd do a quick little sketch, because once you sketch it out, you can see what the effect would be. Yeah, Elizabeth, Asterix again, yeah. You can see if you've not got enough CO2, then you're going to carboxylate less Ruby P, which means you're going to make less GP, which means you're going to make less TP, which means you're going to regenerate less Ruby P. The whole Calvin cycle is going to slow down. Again, you're going to make less glucose ultimately. So it's the same question. It's not straightforward AO1, it's more AO2, but basically it's testing you on your knowledge of the light independent reaction as well as limiting factors. But my little tip, and I do say this to my students quite a lot, sketch out the cycle. Any question that you think is linked to the light independent reaction, quickly sketch the cycle, and then whatever they say, maybe they're talking about, well, there's not enough Ruby P, or there's not enough carbon dioxide, or maybe Rubisco's getting denatured. Once you've sketched it out, you can then go through and describe exactly how it's going to affect the whole rest of that cycle. Ice pass. Yeah, perfect. Perfect answer. Okay. That was my answer, but you've literally typed it into the chat for me, so well done. Ice pass, this will decrease the rate of the light independent reaction. Ruby P can't be carboxylated, less GP is produced, less TP is produced, less glucose, less... You can have my job. That's perfect. Do you have any tips on how to link what the question is to what topic? Ooh. Read the question carefully. There'll be something in there giving you a clue. It might mention water potential, and then you know it's osmosis. It might mention temperature, and then you know it's usually enzymes. Doing past paper questions as well, because you do see lots of the same ideas come up, just in a slightly different context. But it's reading the question carefully, and as I said before, just think to yourself, right, what have I done in class that they could be trying to ask me about here? Because it will be something you have learned you can apply to that question. Okay. Right, will there be a lesson on the light dependent reaction? In January, yes. If you sign up to our January course, literally the first thing we're doing is photosynthesis, and we are going to do a whole lesson on the light dependent reaction, which is, again, quite tricky, but I will simplify it for you. Okay, I will simplify it, hopefully make it easy to understand. That's a really good question. Do you have to say what effect it would have on the plant? You could do, yeah. If, again, you've got to read the question. If the question mentions growth, then they do want you to get all the way to how it will affect growth. So, for example, if this reaction is reduced in rate, you're going to make less glucose. There's a few reasons why that would affect growth. If you've got less glucose, they're going to make less cellulose, for example. Less cellulose means they can't make cell walls. Also, less glucose means less respiration in the plant. If there's less respiration, the plant's going to make less ATP, which is needed for growth. So yeah, if the question goes to growth, if the question mentions the word growth, then try and link a lack of glucose to less growth. So you can say make less cellulose. They use glucose to make protein, so less protein would mean less growth. Less glucose for respiration, less ATP, less growth. So that's a really good question. Okay, let's talk about our course in January. Obviously, if you're here, that's because you really care about the grade you get at the end of your A-level. And there'll be lots of you here that want to get an A or an A star, and that is very hard to do with the limited time you have in lessons in school or in college. So what we can offer you is extra support. So on top of your school lessons, you can join me twice a week for 2 biology lessons. Each lesson is 2 hours long, so in total you get 4 extra hours a week of biology support, biology teaching. You can also get access to your own mentor. Now, our mentors are amazing. They have all got an A or an A star recently in A-level biology, and they are there to act as your one-to-one kind of personal support. If you've got any questions after the lessons, if you want help with your homework, if you want extra questions, revision tips, they can help you. Every lesson that you have with me, I will set you a homework to go with it. And I will also provide you with video solutions to those homeworks where I talk you through exactly how to answer the questions in the best way. You will get a workbook to go with each lesson. So at the end of the course, you'll have all of your workbooks, which will basically form the best revision guide or the best textbook that you could possibly get. They've been written by me and then professionally designed, so they simplify everything. So it's easy to understand, yet still everything is included to enable you to get an A star. You can join our biology community, so you'll have a big network of support with all your fellow biologists who are also studying A-level. And if you sign up for the course, you do get a 14-day money-back guarantee. Just to make it totally risk-free, so you can start the course in January, and if for some reason it's not working for you, you can get your money back within 14 days with no questions asked. The key figures, currently with MyEdspace, we have over 1,300 students studying with us, and 90% of those students recommend our courses. In fact, 82% of them have gone on to achieve, this was last year, gone on to achieve a higher grade than what they expected or what they were predicted. And if you have a look on Trustpilot, or if your parents want to have a look on Trustpilot, there are hundreds of 5-star reviews, and you can read them. They are excellent. If you like the sound of that, put a plus in the chat. If you've got any questions, type them into the chat. Celeste, the course starts in January, but it literally goes all the way up to the night before the exams. So you'll have 4 hours of A-level biology lessons a week, obviously plus your homeworks and video solutions. You'll have extra lessons during the Easter holiday. And you can also get lessons the night before the exam, where we can do some revision together. Okay. The price of the course is £700, which is a bargain, but if you stay tuned, we can offer you a discount. So do stay tuned, because it's an absolutely massive discount, which is incredible considering everything that you get. Which, remember, is not just the 4 hours a week of lessons, but it's the study notes that have been professionally designed. Every lesson has a homework. Every homework has a detailed video solution. You get access to the community of biologists, so you can all support each other and share questions and share advice, revision tips. You can also access my lessons on YouTube. So after the live lesson, you can go back and watch it as many times as you want. And don't forget, you can also get your own mentor who has just done A-level biology and got an A or an A star, and they can help you night or day with any biology question, or they can give you extra questions to kind of extend you and push you for that A star. 44% off is the discount that we can offer you today. So if you like the sound of the course and you want to sign up ready to start in the new year, So we're going to start the course week beginning 8th of January. And as I said, it's 2 lessons a week going all the way from the 8th of January up until your exams. You can get 44% off, which basically takes the price down to less than £400. It works out about £4.90 per hour, which I know if you've had a tutor before, or maybe you've got a tutor now, they charge anywhere between 40 even up to £100 an hour, and quite often you don't get homeworks or revision notes from them. So with this package, not only do you get the lessons, but you get everything else that goes along with it. In terms of specification, the course is going to be focusing on AQA biology. So it's mainly AQA. Having said that, a lot of the content is the same, and I have taught OCR, so I do know kind of the differences between the two, which I'll probably mention during the course, but we are going to be focusing on the AQA, or we're going to be following the AQA specification mainly. A lot of it is the same though. As you know, for example, respiration and photosynthesis, exactly the same. We can split the cost into 2 instalments, so you can pay it in halves, half now and half in January, I think it is. The moderators will clarify that for me. If you want to get the discount, it's valid until midnight on Friday. So the way to secure that is to message us on WhatsApp. The easiest way to do it is to go into the description on YouTube and click the link in the description. It will automatically take you through to WhatsApp, and it will have the letters LIR for light independent reaction. It will have them automatically typed in, so you can just hit send, and that will basically let the team at My Headspace know that you are interested, and they will be able to give you more information and hopefully get you signed up for the course. But remember, the discount is only valid until Friday. So you've got until midnight at Friday, and midnight on Friday, the LIR is just to let the team know that you were in this lesson. So that is the discount code for this lesson. As I said, the link is in the description on YouTube. So go there just straight after the lesson, even if it's just to get more information at this point, or your parents want more information, you can do it by clicking that link in the description. Okay, this is your last opportunity now to ask any questions. So do put them into the chat now. For example, if you miss a lesson, remember the live lessons are stored on the learning platform. So if you miss one lesson, you can watch it back at a time to suit you. Ideally, you would attend them all live, so you can interact in the chat, but we know that sometimes that's not always possible. But you can always access the lesson via the learning platform. In terms of getting enough attention, you can use the chat function during the lesson. You can get your mentor support, who will give you as much attention as you need. You could literally message them every hour if you wanted to, if you had that many questions. And you've also got that community of biologists that you will become a part of, where we're hoping there will be lots of communication back and forth in terms of questions, support, revision tips, advice. You'll also get my video solutions to all the homeworks as well. You can ask me for help during the lesson in the chat. You can ask your mentor for help. You can ask in the community for help. And there's always someone available. The team at My Headspace are always available to support you if you've got any questions. Like I say today, even if it's just questions, you can still click that link in the description and someone will answer your question and give you that additional help or guidance that you might need. Hopefully the lessons will be useful. I'm hoping that you found this lesson useful. I'm hoping that we simplified it for you so that you're leaving thinking, oh, do you know what? The light independent reaction is not that bad. It's actually quite easy. I think it is, you know, just look over it a couple of times now and you'll be a master. But the figures do speak for themselves. So 82% of the MyEdspace students surpassed their goals or at least achieved, you know, the A or the A star that they wanted to achieve. You've got your 14-day money back guarantee. So you can try the course, and if it's not for you for whatever reason, you have got that peace of mind that you can get a refund. So after the stream, we're going to finish in a minute. Go on to the link in the description, click the link, and that will send the LIR, which is the discount code, through to WhatsApp, through to the team, and they will be able to get you signed up or just give you more information if that's what you need. Okay? That's it from me today. I am hoping you join me next week. Next week is our final free stream, and we are going to be covering the nitrogen cycle, which again is year 13 A-level biology. Again, it's one of those topics that some students say, oh, it's really difficult, can't learn it, can't understand it. We're going to simplify it right down, and you're going to go away thinking it's easy. Okay, it's not hard, it's simple. So that's next week, nitrogen cycle. Please join me there, and in the meantime, go on the description and get the information that you need so you can sign up to our A-level biology course in January. Thank you so much. If you message in the chat, how are you able to get the study notes? Someone will answer that for you and make sure that you get the study notes from today's lesson, because they match the PowerPoint, and obviously you can go back through them in your own time. You can even have a go at writing your answers to all of the questions that are on there. Thank you so much. I will see you next time. Thanks for all your interaction and all of your questions and answers. I'm hoping that gave you some confidence. Thanks so much. I'll see you soon. Bye, guys.